Welcome back, everyone. Our next speaker is Dr. Mitchell Ho. Dr. Ho is a senior investigator and the deputy chief of the Laboratory of Molecular Biology and director of the Antibody Engineering Program at the National Cancer Institute. We're really excited to have Dr. Ho present today. His talk is entitled Antibody Binding Sites as Therapeutics, from scFv to VHH and VNAR. Dr. Ho, the stage is yours. Welcome.
Yeah, thank you, Serene, for a very nice introduction. And thank you for introduction. Thank Matt and Serene for organizing this great conference. And so my talk might be slightly different for most other talks. We are going to more focus on the antibody part and how antibody could be useful as the high throughput methodologies for therapeutics, which I have been focused on, but obviously they can be used for diagnostic and many other applications, including as a research tools. And so my laboratory is Laboratory of Molecular Biology in NCI in Ancestor, in Building 37. And welcome if you have, so we're always looking for collaborations. We have collaborated with Matt for many years, and we publish a paper together, at least 2-3 already in the cancer field. So I look forward for continuing collaboration with NCAS and other labs, and including our industry friends here. So the title of my talk, as Serene said, is really about antibody binding sites and how we can use the antibody binding sites as a minimized antigen recognition domain to be used as therapeutics. And so this is the antibody I'm going to talk about. And so antibody is just like it's IgG. It evolved 300 to 400 million years, and it's a really elegant molecules. They have variable regions, they have constant regions, and the constant region has a very defined disulfide bonds to connect the heavy and the light chain together. And each antibody has a variable region and a constant regions. And as an IgG antibody, it has 2 antigen binding sites and could capture like a clamp to the antigens. And the antigen could be virus like SARS-CoV-2, it's a neutralizing antibody, or could be any kind of pathogens or even disease like tissues, like cancer cells. And we have the carbohydrate in the middle, and it's formed, because of that, it formed a conformational, like a ring. If you look on the electron microscope, it's very obvious, you won't miss it. And whenever you see this very signature kind of ring, you know it's Fc fusion, or the Fc part of the antibodies. So we see this quite frequently. And the antigen binding site, and I'm going to talk about, is really about the loop on the structures which binding to the antigen. And the other part is actually the beta sheet of the antibodies. And if you look just by the sequence of the antibodies, many years ago, I think early 1990s, Carbate is one of the pioneers. He's a biostatistician. Just look at the sequence of antibody. At that time, you have hundreds. Now we have millions, even millions of thousands of millions. So now, just looking those hundreds of sequence of antibody, you already get an idea. Some of the regions are hypervariable statistically because you can change it to other amino acid. Relatively flexible, but some other region we call the framework, it cannot be changed so easily because they are very rigid. And so they are in the beta sheet of the antibodies. So what you can change it is the CDR region, that is the antigen recognition sites. And both heavy and the light chain has 3 CDR3, and so that means you have 6 CDRs to recognize antigens. And to be able to isolate the antibodies, there are many methods. I try to summarize. There are only 4 methods in antibody discovery in general. Everybody has been using. The first one is phage display, is our favorite method. So we use this almost every day in the lab. So we could actually isolate B cells from human or animals, from llama, from camel or sharks, humans, mouse, rabbits, put on the bacterial phage, and we can screen them. So I'm going to touch more detail. And the transgenic mice has been developed in early 1990s as well. So you can basically put the human immunoglobulin chromosome into the mouse genome and let mice make the human antibody or variable regions. And in that way, you could just use a typical hybridoma technology to make antibody, but instead of getting mouse antibody, you get a human antibodies. And the B cell immortalization is actually use the virus to immortalize the human or animal B cells to make it stabilize and immortalize so you can select them. And a single B cell is more frequently used in recent years. You can use flow cytometry to sort the single B cells, and you do NGS to look all these sequence of B cells and try to narrow down what B cells might be particularly interesting. So you can use the high throughput FACS to isolate the memory B cells, even plasma cells, and then you sequence them and do those things. So all this technology has been widely used, and maybe the three of them are more frequent than the B cell immortalizations, but all of them you have seen the papers published in the last several months regarding the SARS-CoV-2 neutralizing antibodies. So almost all these methods have been used in these efforts. And so we are cancer lab, so we are interested in antibody, how they may kill cancer cells. And there are several mechanisms. So one of the things we particularly interested in is antibody by itself could be like an antagonist to block the signaling and then disrupt the cancer signaling to be able to inhibit the tumor growth. And one of the examples we have used like GPC3 antibody, we can block the Wnt signaling and reduce liver cancer growth. And some other like Herceptin could also block the HER2 signaling and inhibit the cancer signal. And so this is a direct inhibition just using antibody. And antibody FC part could be involved in ADCC and complement or the other immune response or innate immune response. And this has been used not only for cancer cells, also for SARS-CoV-2. It's not just neutralize the virus, you could also use ADCC or phagocytosis or sensing to inhibit the virus spread. And the other thing is we use now, I think we are actually quite interested in this in the lab is CAR T cells. So we could put the antibody on the T cells to use immune cells like T cell or NK cells or other immune cells to deliver those immune cells to the tumor microenvironment, kill the tumor. And so antibody in that case is like a guide for the immune cells to get into the tumor microenvironment. Otherwise, it will not be infiltrated or may not be there. And so we could also use ADC or immunotoxin to fuse the antibody with toxin or small molecules. And we also do bispecific antibody collaborating with, in that case, we are more collaborating with companies to do the bispecific antibody to try to attract the immune cells to the tumor cells. So there's really multiple strategies to try to use antibody to actually kill the tumor cells. And so hybridoma is actually has been developed in 1970s early by Kohler and Milestone and in Cambridge. And so that is actually the Michael Porter in NCI has contributed significantly to this field as well. And he is one of the first lab showing the myeloma cells, for example, mouse myeloma cells can produce a monospecific antibody. Actually, now we call monoclonal antibodies. And so if we have rabbit or mice and we fuse so we can immunize those mouse or rabbit with the antigens. And we can do the fusion with the myeloma cells. And then we could do the so-called hybridoma technology. We can actually screen them. And usually we use ELISA and we select the clones. And in my own lab, we actually developed a so-called high throughput screening of hybridoma clone protocols. And that is many years ago when we were the first, one of the really first lab. There were only dozens of labs in the world and actually bought this machine at that time. So it's called Guava. It's a very premature kind of 96 well flow cytometry. So we were the first one to try to use that to screen antibodies. So now they're the more advanced and updated machines. Even the core facility, including 37, has another one. And back to that time, we used guava, and we mixed the positive and negative cells and at a high throughput, so to see which antibody or which hybridoma actually bind the positive cells, a very strong signal, but the negative cells with the least signal. So we mix the positive and negative cells in each sample. And particular look, the antibody can bind to the positive one very strong, but for the negative human cells, which is normal cells, and so they are not binding at all. So in that way, we could screen the hundreds of solvents. Actually, for mouse hybridoma, we screened the 700 to 800. For rabbit hybridoma, we screened 7,000. So in this kind of technologies. And by doing that, we actually successfully screened the two human antibodies called YP7 and YP218. Now they are very widely used in many labs in the world and to target the GPC3 and the meso-3 for bispecific or CAR T cells and among others applications. So those are really highly tumor-specific antibodies. In the way we select them, we screen on the so-called high-content cell binding assays. And after we get a mouse or rabbit antibody, my lab has put a lot of efforts to try to do protein engineering to humanize them. The one way we do that is we compare the antigen and antibody complex in the PDB database. So I remember we have like not many, like for rabbit antibody with the antigen, we probably have dozens of them in the PDB, not hundreds of them. So we look them each one by one and to try to understand what the CDR region I'm talking about actually involving antigen binding. So we decided based on those structural analysis and what amino acid, for example, within the 20 angstrom or 15 angstrom might be the direct contact, or maybe 50 angstrom is not direct contact, might be important for folding or sensing. So we, based on this, actually we grafted the CDR and to the human framework. For human framework, we also look at the germline sequence to make sure which one might be the most dominant in the human and which one might be more likely to fold very well. And as we see many papers published for COVID-19 neutralizing antibody, you see that VHH, for example, is a dominant germline family in any neutralizing antibody in different labs or even in different strategies. So you can see germline sequence in human are not random, so they have some kind of pattern. So we particularly look at those germline sequence and grafting the CDR into the human sequence. And we successfully humanized the humanized YP7 and the humanized YP218. And both of these humanized versions now underway for clinical trial as a CAR T therapy. So that is quite exciting. Yeah. So for humanized YP7, we are collaborating with Rafi Hassan's lab, trying to use it and funded by Cancer Moonshot program to actually test in the mesothelioma or maybe lung cancer or ovarian cancers. For humanized YP7 CAR, we collaborate with Tim Gratton's lab, also in NCR Clinical Center, try to treating liver cancer. So that trial could be started very soon, maybe early next year. And to be able to minimize the antibodies, so what we talk about so far is IgG antibodies. So how we can minimize just the antigen binding site, because that is something why I'm going to talk about why we are interested in doing this. Because for protein engineering point of view, we want to make it smaller. We want to make the fold well and express well. The whole IgG is very difficult to express, for example, in E. coli, but a single chain V will be easier to express E. coli. But it will be even more easier to express E. coli once we talk about single donors. So this is single chain FV, and Jim Houston is one of the first actually proposed that might be a good idea to have having a light chain and with a flexible linker, usually called the G4S linker, and there's a 15 amino acid and to connect the VH and VL. And this is Jim Houston's earliest paper in PNS, and he actually presented first the VH followed by VL, and you have a flexible linker. And now we call single chain FV, and you could express in E. coli. That's the key. And it will be easier to screen because you cannot put the IgG on the phage virus, the bacterial virus like a phage. And you can, so later on we could also put single chain V in yeast cells or mammalian cells relatively easy because it will be more difficult to put a single chain V on phage or yeast. And so the phage display was originally developed using peptide, and then ultimately a single chain V can be used to fuse to the phage. And that's a very important development. And then we can easily express the single chain V as a bivalent, for example, BITE, with bispecific T-cell engagers. And we could also make immunotoxin to fuse the bacterial toxin and produce E. coli. So I'm going to touch a little bit about that. And later on, we can use single chain V to make a CAR T cells. And for single chain V to express on the bacterial virus, it does, this is the M13 phage, it's very long filamentous phage. And on one end, you have a P3 protein. It's actually had 5 P lines, and that's actually what phage used to infect the bacteria. And at the end of the P line, the P3, so you are going to fuse single chain V to it. So because single chain V is longer than peptide, but it's still relatively small. It's about 25 to 30 kilodalton. Actually, it's just the right size for that to display on the bacterial phage on the P line P3. And this is a typical phage display. And each of the bacteria, they will have a particular vector express particular single chain V. So the library size could be 10 to the 9 to 10 to 11 or 10 to 12. And so usually you try to depending on the antigen on the immobilized surface, like immutube or Lassa plate or whatever, or magnetic beads. And once the phage binds to the antigen, it will be actually attached to that surface, and all the other will be go to the trash can. So you do this actually 3 rounds or 4 rounds, you eventually will enrich the whatever binds to the antigen immobilized on the surface. Okay, and by doing this, and besides the single chain V on the phage, Dan Wittrup, when I was a graduate student in Illinois, Urbana-Champaign, and Dan Wittrup was assistant professor over there, and he was the lab actually developed yeast display back to that time, and that turned out to be a really important development because yeast is same like E. coli is very tough, but on the other hand, you could do Phage because it's big enough. And so that will make it easier for screening and using flow cytometry. And later on in NCI, actually, we developed a mammalian cell display. So basically, we just put a VH and a VL as a single chain V and on the cell surface. But in that case, we use a PDGFR transmembrane domain to anchor the single chain V on the cells. So we're still using developing mammalian cell display in the lab, just use a different kind of context and different kind of molecules or different kind of libraries, including single domain library in the lab. So we're still doing that. And for immunotoxin, we will try to put a single chain V, and which is also from antibodies, and to fuse a bacterial toxin. So we call domain 2, domain 3. Domain 3 is yellow, domain 3 is red. And we remove the blue part because that's the binding side, and we don't want it. We replace the binding side with the antibody single chain. So, and we put a flexible linker between, and so that will separate the 2 domains. And we have an antigen binding side, which is single chain V, followed by a flexible linker. Then the toxin part, and that is toxin domain 2 and the domain 3. At that time, we believe the domain 2 is more related to translocation, and the domain 3 is the key. That's the functional domain. That's the enzymatic domain. That's how PE actually works to inhibit the protein synthesis. But later on, we find that we don't actually need the domain 2 at all, at least for some cancer type or some cancer cells. So we actually even remove the yellow part to keep only the red part. And that's the functional domain of the bacterial toxin. And in the laboratory molecular biology now, and so we have developed the moxetumomab, the toxic anti-CD22, and that was approved actually by FDA 2 years ago. And this immunotoxin is actually binded to the CD22 on the leukemia cells, and it's internalized. Most of the immunotoxin will go to the endosome and ultimately go to lysosome, like a trash can, and be degraded. But some of the immunotoxin will survive and they will eventually go to the trafficking to the ER. And then during the ER, they will actually translocate to cytoplasma. In the presence of NAD, there will be ADP ribosylation of the elongation factor 2, actually in the protein synthesis machinery. And in that way, it will inhibit the protein synthesis. And ultimately, some of the cells will die. And because they induce apoptosis, you will see the drop of the MCL1, for example, pro-survival markers. But some of the cells will die. But not all the cells are die. Some will be resistant, even though protein synthesis is inhibited. And that turned out to be something very interesting to study. And that's why we actually collaborate with even ongoing project. We collaborate with Matt's group, actually try to use small molecule to combine with immunotoxin to see if we can sensitize the cell death in this step, and to be able to use together small molecule and immunotoxin to sensitize some tough tumor like a pancreatic cancer. They're usually resistant to almost everything, and the liver cancer as well. And so we actually published a paper on liver cancer. Now we are doing on pancreatic cancer as well. And to be able to use both small molecule and immunotoxin, they are using different kind of mechanisms. A lot of small molecules, DNA, like damaging agent, or maybe microtubule inhibitors or others, but the immunotoxin has a very novel mechanism of killing because that inhibits the protein synthesis. So it would be good to combine. And for CAR T cells, many years ago, and Zadok Ashkar and others in late 1980s or early 1990s was one of the first lab think about maybe we can use TCR and replace the variable region of the TCR with antibody like single chain V to make a chimeric TCR. So we'll kill the tumor cells independent from the MHC. So that would be a good idea, right? So it's intuitively would be very attractive. And but the problem is to make a TCR like recombinant complex would be super difficult. And even now, today, if we want to do that, I think we and the other lab are still trying to do it. And but I think the easiest way to do it, as they found that in early 1990s, if we could put a single chain V fused with CD3 that would be the easiest way to do it. So they fold well, they express well in primary T cells, but the efficacy was not so good. So then later on, they put a co-stimulating factor like CD28, 41BB, and others to make so-called second or third generation CAR. And in our lab, we actually put a single chain V, like what I talk about, the humanized YP7. We humanized YP7 use single chain V and targeted GPC3, that is liver cancer. And on top of that, we put a human EGFR truncated form, just the binding site for cetuximab. So then we can use it to monitor or label the CAR T cells. On the other hand, we can remove it if we want as an off switch. So this is what we think might be very useful to make a CAR T cells using single chain V, humanized the one we made. And like any research, when we're doing the research, we always find something surprising to us. And when we talk about the GPC3, making GPC antibodies, we found another nanobody. At that time, it's engineered human nanobodies. It's a man-made antibody. It's not nature occurring antibody, but it's nanobodies in the phage. And they do not need a light chain to pair. So usually in human, you need to have a light chain to pair IgG. But even in humans, the heavy chain disease, there are some heavy chain diseases, those patients only have a heavy chain antibody, do not have a light chain. And they still survive. It's not lethal, but the problem is they just don't have a light chain to pair with their antibody. So the heavy chain antibody disease. But that means that some of the human antibody, even the germ, depends on the germline sequence and others. They don't have to have pair with light chains. So that's what it is. So we have a human antibody, and we find that the nanobody could actually bind to the Wnt binding site. So it took us several years to figure out where it binds. And this is EM. We find it's validated what we find. It's actually indeed bind the end lobe of GPC3. And we also try to solve the Wnt binding domain in these regions because it was a surprise to us because most people believe Wnt, like other growth factors, will be attracted by the heparan sulfate chains, but nobody realized they could actually bind the core protein as well. So we use this H3 antibody to show it could block the Wnt binding on the core proteins. And indeed, to map this binding role. And using this antibody, we actually published a series paper to show this antibody could actually block the Wnt binding site and eventually reduce the Wnt signal in the liver cancer cells. In addition to the toxin part, immunotoxin could inhibit the protein synthesis. So the dual function of this immunotoxin become very potent and better than any other anti-GPCC immunotoxin in our trial. And so this is something we think will be very useful. It's not only for immunotoxin, maybe for ADC. And we start to believe why the TDM4, like trastuzumab ADC, works so good and many other ADCs not to be not so successful, is part of the reason the antibody part is not as functional as a receptor. So in our case, we find that the nanobody here, you can block the Wnt signal, play a very important role to make this immunotoxin very potent. So I think this is a very interesting observation, and because of that, we actually continue engineering them. And one of the more recent engineer actually published a paper with the Matt Foss group in this paper. So we actually engineer this immunotoxin, and including the nanobody and another nanobody targeting the albumin. Okay, so we have two nanobody here with a flexible linker. So in this case, it's a bispecific nanobody immunotoxin. So we can increase the half-life because they bind the albumin. But on the other hand, we also remove the T-cell epitope on the toxin, so we can potentially make less immunogenic because it's bacterial toxin. So in that case, we optimize our immunotoxin. It has a bispecific, has a longer half-life, and less immunogenic, and hopefully we can use for, so we can put them into the clinical trial, treat the liver cancer patients. So this is how we actually discover nanobody could be very useful. And we probably one of the first group actually identified the nanobody could block the signaling complex, in this case, Wnt signaling. And so that's actually very exciting to us. And because based on this observation and the other project done by Nandi in the lab, and she actually used the nanobody and to see it actually could downregulate the Wnt signaling for another target called GPC2 in neuroblastoma. And more striking is you can actually reduce the expression of N-Myc, and that is the targeting of the Wnt. And N-Myc is believed to be an oncogenic driver for neuroblastoma, and downregulate N-Myc because most neuroblastoma has N-Myc amplifications. Downregulate N-Myc turn out to be very important therapeutic effect, and that's what nanobody could do to block these Wnt bindings. And because of that, we start to invest more about the nanobody in the lab. And so we get some innovation grant from NCI and also NIH to start to work on this. And one of the things we actually find very interesting is that's in nature occurring nanobodies. And for example, in Camel, and there's a heavy chain antibody like this. And in shark, you have a heavy chain antibody like that. So both of them, they have a so-called single domain, which is only 12, 13, 15 kilodaltons. It can be used by itself. It's a functional domain. And a single domain has been evolved at least twice. So I say at least is because I believe it's much more than what we know. Because even we are in the era of genome, we don't sequence all the genome so we usually sequence the genome in the model organisms, not those non-model organisms. So if we sequence a lot of non-model organisms, we're going to find probably more and more and different kind of formats of antibodies. And so for heavy chain antibody, and so we already know, in addition to shark, like a ray or skate, they also have heavy chain antibodies. And for camels, the alpaca or llama or camels, they all have some kind of heavy chain antibody in the camels. So it seems like they evolve, and the heavy chain antibody has some advantage during the long period of evolutions during the animal kingdom. And about 400 or 300 million years ago, and that is the age of a fish. So basically, there are really a lot of sharks or different sharks in the oceans, and in that, and we still have more than 400 species of sharks on the Earth. So maybe many of us may not be aware of that. So we still have a lot of different species, and back to that time, we have even much more. And And when shark has the earliest adaptive immune system on the Earth, because they also have T cell, they have MHC, they have B cells, they already have those basic elements of the adaptive immunity. And same as, of course, amphibians, reptiles, and birds, and of course, mammals. And so shark is really the earliest adaptive immune system. And what is amazing in shark is, and not only the B cell receptor I just talked about, they have heavy chain antibodies. So they have a single domain we call VNAR, is NAR. And even in the T cells in the shark, they also use VNAR as antigen recognition site. So that means that VNAR and can be used both in B cell receptor and the T cell receptors. That is very fascinating. That means in shark, in the fish, so these variable regions can be used both by B cell and T cell, attacking antigens which do not need an MHC or need an MHC. They use the similar kind of variable domains. And because of that, and so we found that might be very interesting to work on the shark and the camel single domains. So overall, the nanobody has some advantage I try to list here. They could bind to the buried functional site and not accessible by conventional antibodies. So they could bind to the cavities, and which the conventional antibody may have difficult to get in. So I'm going to show you one example. And they could actually bind to the glycan. Some glycan will be totally not immunogenic, such as heparin. And you can extract heparin from the pigs and put into human, has no immune rejections. And you can, the nanobody could actually use as research to stabilize the membrane protein like GPCS ion channel for structural biology. They could also isolate a cross-species nanobody because they really don't care about the primary sequence. They really care the cavity, the conformation of the protein. Then, of course, they could be virus neutralizing activities. They could easily produce the E. coli insects. They could, of course, do multi-specific, tri-specific already. And for CAR T cells, their transaction efficiency is just far much better than single chain because they are easy to express. So one of the first nanobody already approved by FDA for blood clotting disorder, so it's used for infusion. But nanobody could be also used, potentially used as inhaled drugs, or maybe as like sprayed or eye drops, because they are very small and soluble. But the first one approved actually is for the systematic treatment with the blood infusions. So the nanobody could bind to the cavity of targeted protein. It's not only just because they are small. They also have a loop that is like a bait. You have the different number and the location of cysteine in the CDR3 that may be important for them to get in the cavity and bind to there. And that is kind of missing in the conventional antibody. They usually bind to the target relatively flat. And there are 4 types of shark single domains. And the human and mice mostly have this kind of the antibody we call type 4. And the canonical pair of the cysteine connect the CDR1 and the CDR3 just right before CDR1 and CDR3, the framework. And that's between the beta sheet to stabilize the variable domain. And for shark and camel, even in camel, so there are a lot more about type 2. That means you have an extra pair of non-canonical cysteines to connect CDR1 and the CDR3. So you see that the bridge between the CDR1 and the CDR3 to make them actually together as like a complex of the structures. And for shark, particularly for shark, they have even more CDR non-canonical pair of the cysteine. So in this case, they connect CDR3 and the framework, and so make a very, very unique so-called bicycle structure, which you never see in any other antibody. So that is unique in shark antibody. So you have this kind of bicycle shape in the CDR3 loop. That is the antibody binding site. And they have really a unique binding site because of the bicycle kind of shape. So for type 1. And so for several years ago, we started to make antibody, single chain live only. And so this is a shark live only. We get 6 nurse shark from the local Atlantic. And so those sharks, so we get the blood. And as I told you, we want the B cell, T cells because they share the same VNARs. and we PCR amplify it into the phage, and we express them. We do NGS to make sure that the diversity is about 10 to the 10th or even more. And we use this library to do the screening. And later on, we also make 20 Camel nanobody libraries. And we made, in this case, we made each Camel library like 10 to 10th phage display library. And the Camel actually has the age from 3 months to 20 years old, and 10 female and 10 male. So these Arabian camels, so we actually get it from not in Asia, but actually in the Midwest, in the US, actually Missouri, from the farmers. So we actually get those blood, and so we make all these library. Again, we also confirm diversity by NGS. So we have those made those very big shark and camel library, and basically we have the largest such library in the world. And so each library have 10 to 10. So overall, we may have 10 to 11, even close to 10 to 12 diversities. So in that is a very useful resource. And then when we do NGS, we could find that the CDR region have a multiple cysteine. So I highlight the cysteine here. You could see there are multiple cysteine, unlike human mice. They only have these two canonical cysteine highlighted in yellow, but a camel and a shark, and a particular shark, you have multiple cysteine in different regions to make a very different conformation, very diverse in terms of conformations. And we could make it in E. coli. So those so-called type 1 or type 2 shark, and so they have multiple cysteine, they have more cysteine than human or mouse antibodies. We could still express them in E. coli. And for the best sequence, we could get like 200 milligrams from 1 liter, but we routinely could get like 10 or 20 milligrams from 1 liter, and to make those in E. coli. And it takes about a day or two, so we could make 3 or 4 or 2 or 3 nanobody in E. coli after screening. So in the last about 5 minutes or 7 minutes, I'm going to talk a little bit about how we can use this nanobody to do something actually very fascinating. And so we have made a nanobody to to cancer antigen, but I'm not going to talk about today. So I want to particularly talk about using nanobody to target the SARS-CoV-2. And especially our next speaker is talking about SARS-CoV-2 also. So I think that would be very suitable to just put into the one session, and we are focused on this today. And so for the nanobody, and we have a nanobody, so we try to block ACE2 and the spike protein from the SARS-CoV-2, and we believe nanobody is small and they're conformation sensitive. That would be a good application for that. And we also try to get a nanobody even bind to the fusion stage, which nobody has been able to have a good success on that. And so we thought that maybe nanobody is small enough to get in the fusion stage and block them. So that's something we think it might be a important role using our nanobodies. So Jessica in the lab actually used the 6 of our nanobody library from Camel, 3 male, 3 female, as I said, from 3 months to 20 years. So we actually on purposely just try to include the different age, different gender camels into our screening. During the March to June, and Jessica just screened in the lab and try to use all these libraries one by one, and each of them related to multiple rounds and different strategies. So I'm not going to go to detail. So basically, we're actually planning on the RBD or spike protein of the trimer of the SARS-CoV-2 and try to get those nanobodies. So this is probably the crowded slide I have, but that's probably the most important one in this part. So we actually have like 700 binders to the SARS-CoV-2 spike protein, and we actually sequence 100, 200 of them. We finally narrowed down about 29 nanobodies that actually bind to the, not only just RBD, but also trimer. And one of them actually bind the SARS-CoV-1 as well as SARS-CoV-2. So that might be particularly interesting. So we call the group 1. And or the other group, they are particularly just binded to SARS-CoV-2. And to be able to validate the group 1, that also bind the SARS-CoV-1, and we also make a stable line, which express SARS-CoV-1 trimer, because at that time, we don't really have a trimer. We use S1 of SARS-CoV-1 to do the screening and planning. but we want to make sure. So we make the trimer stable line and in the lab on human cells, and we show the SARS-CoV-1 the binders like 1B5 actually binds the right spike. And so we have the control like CR302, which also binds the SARS-CoV-1 spike. And that's the only nanobody we have to cross the both SARS-CoV-1 and SARS-CoV-2, but all the others, they bind only SARS-CoV-2. And when we look at the sequence, I actually try to explain that we are very interested in the, what the type of the nanobody we have. We find about 6 top binders and the top actually the inhibitors as well. And among the 6 of them, 4 of them are type 2. So that means they have additional pair of the non-canonical system to connect CDR1 and CDR3. So in that case, you will not find such antibody in human or mice because that's unique for camel and shark. And but we also have the other 2, which like a human mice, so they like type 4, they only have the canonical pair of systems. So overall, we have 6 good binders and also the good inhibitors. And when we test on pseudovirus, so this is the pseudovirus assay. So I just show some of the data. So we could actually see the 7A31B5, that's the one cross both SARS-CoV-1 and 2, and this. So we could get a low nanomolar IC50 to inhibit the pseudovirus. And if we do the cocktail, just combine 2 out of them, and we are continuing to do that because we have 6 top binders, and we could go down even lower, like 1 or 2 nanomolar to inhibit the pseudovirus. So we thought that might be very interesting. And so we continue to testing with our collaborators on live virus and animal protocols. And so hopefully we can select the top 1 to move to the clinical. And when we do the structure, so, we particularly look for example, the 7A3 they actually inhibit the SARS-CoV-2 in the neutralization assay. And we looked at, and we are still doing ongoing the cryo-EM. And at this point, we just mapped the epitope based on the peptide mapping and others. So we find our nanobody binds to the site which directly interact with ACE2. And while the other 2 nanobody people published so far is called, one is called H4, the other is VHH 72. They bind a totally different site as compared to what we have. And so that might be interesting. And we are also doing the structure to try to understand more. Okay, so this is what I want to talk so far. And I think overall, we have developed all these nanobody technologies in the lab, and we actually developed protein engineering, antibody engineering over the years, some funded by intramural program. And some of the clinical trial we have luckily funded, actually 2 of them funded by Cancer Moonshot program, and some of them funded by Innovation Award from NCI and also N-Myc. And for the COVID-19, we get the COVID-19 award from the NIH ITAC program, which is very good. So we can be able to do that even though we are mostly a cancer lab. So we are able to do this in the next several years, funded by NIH grant. And of course, we have many student fellows have contributed to our program. And we have fantastic collaborators, including NCATS and many other institutes and labs. So for both cancer therapy and emerging infectious disease like COVID-19, so I think the research need a multidisciplinary team from structural biology, cell biology, molecular biology, protein engineering, antibody engineering, and computational biologists and chemists. So we really need everybody to the table to work together and share their expertise so we can move the most exciting science forward and benefit the patients. So with that, I think I'll be happy to answer any questions you may have. Thank you.
Thank you very much, Mitchell, for that wonderful and really fantastic presentation. I'm gonna check whether there is, I don't see any questions in the chat box. So if any one of the attendees have questions, please type your questions in the chat box to all panelists or in the Q&A. Meanwhile, if any of the panelists have questions, please unmute yourself and go and feel free to ask your questions.
So Mitchell, I had one I put into the chat box there. It's a bit tangential to what you were talking about. So MERS lives in camels, and you've been working with camel at antibody libraries. But a lot of the neutralizing antibodies for treating COVID have come from patient antibodies that have been identified in convalescent patients. Does anyone, do you know if people have, or if you could look at camels that have MERS and to fish out the antibodies that they've developed as part of their immunity to MERS to see whether they'd be useful as neutralizing antibodies? Because that's a coronavirus that jumps from camels to humans on a semi-regular basis.
Yeah, no, Matt, I think that's a really good thought. But I always thinking about it in that way too. I just don't have evidence to show whether it's true or not. But you are right. So camels has been shown at least for one of the coronavirus as an intermediate reservoir, and they don't get sick, and they may have a good scaffold and to neutralize the coronavirus, including SARS-CoV-2, and maybe that's why they don't get sick. And so that may be why we actually get a lot of good antibody actually. From this camel, they are naive camel. They are not. They are like antigen naive camels. So they never exposed to SARS-CoV-2 as far as I know. Yeah. But again, and we could find pretty potent, right, the neutralizing antibodies from these camels. So maybe their reservoir or their scaffold are very suitable to attack those coronavirus. Maybe they have encountered for many years, and so they have the right scaffold in their system to attack those coronavirus. I think it's really a good thought, and it's possible. Yeah, and one advantage of using nanobody in this regard is we hope we could make, for example, inhaled drug instead of just infusion. So because they're soluble, they're stable, they probably don't need a -70 or -80 or even -20. So they can be stable in the room temperature. And so that'd be nice, right? So you could put it in the CVS pharmacy and spray it, and, yeah. So that's something, might be, advantage.
Thank you.
Thank you very much, Mitchell. I do not see any more questions coming in right now. So again, thanks very much.
Thank you.
You're welcome.